أعوذ بالله من الشيطان الرجيم. بسم الله الرحمن الرحيم. رب اشرح لي صدري ويسر لي أمري واحلل عقدة من لساني يفقهوا قولي. السلام عليكم ورحمة الله وبركاته. Brothers and sisters in Islam, we are here in week 9 of BIOC17. Subhanallah al-Azim wa Rabb al-Kareem. May Allah accept from us, Alameen, Alameen. Tonight, our students are going to be looking at or preparing, in fact, different types of media for bacterium in different differential forms of media. Inshallah ta'ala, I look forward to talking about this saving time. Inshallah ta'ala, in this anything practical. Inshallah ta'ala, a few more. Hi, Dr. Brent.
We will use this for just the ozonic concentration. So they will evaluate that by using a spec reading at the same spec reading they use for the generation farm. Other than that, for the temperature and the pH, you want to set up one big set of data because they each did different parts of the experiment. Make sure they totally resuspend. But I want them to think about what kind of data they would get if they did a spec reading. It's just a little bit too much work, I think, with them having not prepared for it to actually do all the spec readings. So I want them to pay attention to also getting those counts for the thermal death time, because they have the 0 to 5 minute counts, which you need to collect. And then next week you'll interpret the thermal death time data. So all the data that each TA put up, plus the viable counts. Okay. Because there'll be more time next week. Because of course, next week, all they'll be doing is interpreting the differential media, thermal death time. There's a reflective practice. There's no setup because next week's the last week of labs. Keep in mind that we still need to go through the so-called virtual labs, the Snyders and the Blood Agar, to make sure they understand it. Okay?
Yes.
Sound good?
Sounds good.
Okay, got any questions for me?
No.
Right. I believe that Daniel's saying this culture for the biochemical next week.
Yes.
So normally we would have the culture and we can test this week for that next week.
Check the oh, that's the spectrometer. People were using that part, that. I didn't write that part. There was nothing on it. Okay. No, science the alternative. Yeah, alternative special. Right? So I see that. I know. I knew you were gonna write it. So I was like, why is there two ways to do this? Yeah, battery was the. Me too. What is the visual observation? Oh. I don't think I wrote. Oh, which one? I got you did. Oh, you found it? That's the word that I was trying to use. And fail. Oh, No, 8. Oh, 45. I'm sure it's 10 more. You have on my oh, second one. This is my microbiology. More 20. Okay. Friday, yeah, I go right. I go right out. How do we have? Oh, so stuff to do. Did die? That was too much fun. I think Kaylee is like, you're so funny. I think she like, I think the next morning she was like, you're not too much fun. Like, sure. Guys. Bring that next week. The first time ever. Right back in an hour. Our, we got a like realistic, like that, for fun stuff. Oh, that's cool. Don't feel. Wait, you mind waiting like another? Like, why? I'm like, yeah, seriously. Yeah. And then I was debating between this one or this conch, but I got on this side, but it failed. Yeah. And I had to take it out. And then I was like, can I try the conch? I think it then I was like, I'm not seeing this last time. I saw that. Yeah. It was about but I am coming. Yeah, I should it. I'm looking for like the big ones that are there's an object.
Right. You got to see it. It is going. Oh yeah, this is on. Yeah, you don't want it. Follows out. You know, like. And you're just like. Okay. Oh, yeah. Yeah, I would love to do that. Like, fine, but I have to go to the bus for money. No, I don't feel like you're judging the liver. Can't donate shit on here. The way the so no, yes, generated some it's like because it's all black. Yeah. We're gonna exactly. I'm saying $10,000 in the US. It's like $12,000 Canadian. Yeah, I do. But then Korea's like like it's like I think about it, you should really consider like you should really think about this. Okay, just go ahead and tell your right. It looks like that's like their 14, so like and they're like they like sign up. Yeah, they were. And now I have to do like a question. Like I might be I know that. That's like genetic sequence too. You're not for being as far as that.
Can you please turn on your spec 20s?
I just feel like
You cannot spawn
it's a matter
and we need to have them essentially at wavelength 550 nanometers. So if you can adjust it according to that like, Okay, everyone. How are you all doing? Are you guys doing okay? Congrats. You're here in week 9. Folks, just a kind reminder, make sure that your lab coat is buttoned up. Make sure that you're not using your phones. Make sure you're paying attention. We're here at the finale, week 9. So I hope you guys really come to appreciate what you've done thus far. different techniques from staining to microscopy and other technical skills, which you can start to think about in prep for upcoming practicals. And so we'll talk about that in due time. Now, first off, we're here in lab 9 once again. I do want to highlight before moving forward, some of you may have been missing specific figures for your post-lab this week. So please make sure that if you haven't shown them to me, or if there's anything outstanding, please make sure that you show them to me, those specific details before you leave today's lab. So that way it's marked complete. In terms of other things, lab performance, if some of you are wondering, it pretty much reflects your acuity or your performance essentially in the lab, being able to ask questions, answer questions, being able to complete your tasks efficiently and coordinate as a group when needed. But those were just some things, as well as FLRs. So pretty much expect Dr. Brunt to release them when she's ready. But hopefully, I'm expecting within the week, this week or the following week, that your results will be essentially uploaded or at least published on Quercus, so that way you can see your marks. I did send, by the way, a Quercus. I know it was a bit late, but a Quercus message yesterday. Hopefully that'll help as well for your prep for the UV FLR coming up soon. So just bear that in mind. If you have questions, let me know. But we'll move straight ahead. There was one other thing I wanted to say. Yes, so if you haven't grabbed your unknown assignment, we still have them there. I do want you to make sure to grab your unknown assignment back with the data sheet and flowchart. And if you need to look at the slides, you can. They should still be at that box. And likely, my goal is that if I'm done, inshallah, any other assignments in like a quicker than expected fashion or manner, then I'll try to let you know accordingly. It just takes a lot. Takes a lot to mark different FLRs, but nonetheless, I hope you guys come to appreciate the general feedback or the mini rubric that I provided, because it will be very similar for your upcoming FLR. Any follow-up questions, folks? Any questions? No? But I will say that you guys are keep it up, You're almost there. It's exciting. But now this is where I was actually talking about integrating things back from the past. So we looked at different stains, different tests, looking at different characteristics of bacteria. Now we're doing it on plates, and it's going to be a bit fun because, as you can see across your benches, you'll have 3 sets of colorful plates with different media. But before we go there, you likely know that there are different components of media that have been mentioned in the manual. And there are 2 different categories, selective and differential components of media. Does anyone know? Or maybe should I just say it for time? Okay, if I were to ask, actually we'll talk about it. This is perfect. So let me just go with an intro though. These all reflect, so even the tests that you've done in the past for staining, let's say gram-positive, gram-negative bacterium, endospore, capsule, etc. They have different they reflect different themes or different topics. But in this regard, what you're doing for analyzing like the catalase test or the cephinase test, you have different biochemical analyses at play. And so for these plates, what you're going to be looking at next week for your final week is being able to identify a bacterium with their specific properties of interest. So what I mean is, as we talked about, there's certain enzymes that exist in specific bacterium. We talked about cephinase or beta-lactamase. We have catalase, but there are other ones that you're going to see from the upcoming slides that will likely give you an indicative thought as to what different components do these plates make up and what you would likely expect for your results next week. Yes, so I also have the results of the tubes, and this is where things come up a bit. Let me see. So we'll go first with Lab 9 material, but then we'll come back to Lab 8, like the post-lab, with the results of the environmental conditions, pH, temperature, osmolarity. They should all be in the middle bench, either on that side or this side. I just wanted to separate it from the ones that you have there for Lab 9. But pretty much each bench should have 3 sets of plates, focusing on a Mannitol Salt Agar. For sure, we'll say exercise 1, exercise 3, and exercise 5. DNase test, the test tubes are there, and each group will have 3 reps, three different tubes. One will be B. subtilis, one E. coli, and one control, i.e. no inoculation. And you're just dipping in halfway into the agar. Don't go all the way in, but just halfway into the agar itself. And then, yes, 1, 3, and 5 are the plates that you have. So that is, and you'll see the different colorations. The McConkey Agar plate, which is like a faint reddish pink. You have the Mannitol Salt Agar plate, which is more red, I believe, and separated into 3 sections. And then you have the blue one, that's the DNA hydrolysis plate. So let me just highlight this for you guys. As you look through the manual, you come to appreciate that there are 2 different types of classes for these components. There is differential and there's selective. If you have a selective component of an agar or medium or anything really, you are trying to separate essentially bacterium that would grow versus bacterium that wouldn't grow, or bacterium that would survive or not survive. So a simple example of this is like, as we'll see hopefully from the different ingredients here, and even from the past, we recall trying to think of other examples. Okay, so an example is, if you recall the plates that we use for Lab 6, LB and AR. That was your 4th one for transformation. And the idea is that ampicillin, right, is an antibiotic, as we recall from week 8 as well. So you should know that if bacterium grow, right, following inoculation, then that would be an indicator of a selective component of your medium. Meaning, so if you did inoculations, and then incubated for 24 hours, if you found bacterium growing, then you would know that they're resistant, right? They are resistant. So the idea is that is a selectable marker, right? We call that probably Bio B12 or other B-level courses. But the idea is that if it's if a component is selective, then it's essentially separating a bacterium by a specific character, i.e. if it's positive or negative. Whereas if you have differential components, they may be able to grow, but they have different traits, or like essentially they have, they're more like on a spectrum. So for example, some bacterium are able to essentially metabolize or ferment. Let me just see if they have another example here. I want to go to another one. Yeah, so let's go to McConkey Agar. So essentially the idea is that you'll have the different colorations that exhibit that they ferment their metabolites differentially. And so to make it easier for you guys, if you see like salts Salts are generally, as we can tell, differential because essentially you're gonna, selective, in the sense that if, as we recall from week 8, there's some osmotolerant and non-osmotolerant bacterium. So the idea is that if they're able to grow, then that means that that if your bacterium is able to grow, then it's successful and it's osmotolerant. But if it isn't, then it's non-osmotolerant. And so it's binary. It's like it either is or isn't, and that's how it's selective. Whereas if it's differential, like the case of metabolites, it could still grow, but you'll likely see a difference in color. And that's where you'll likely see it from, yes, McConkey Agar. It's more easier to tell. I don't know if you can see it from the slides, but the difference in colors, that's more darkish pink and a faintest orange or yellow hue, and then the more pale as shown in the middle, quadrant 3. Does that make sense, folks? Are there questions regarding that? So, like, what you're going to do today is essentially look at the different components, right? So for, let me just reiterate this. If we go to the McConkey Agar plate, you have crystal violet, right? That is what, a selective or differential marker? Differential or selective? Selective? Think of it like a dot. Think of it like your gram stick. French. Differential, does that make sense, folks? Yeah, okay. Lactose, it's a metabolite. If bacterium grow, what do we expect? Or actually with including lactose, think of it like a sugar, right? Is it gonna separate bacterium, or is it gonna, like, i.e. be selective? Or would you be able to differentiate bacterium that are able to metabolize lactose or not? Like essentially, could it survive with any other components in the plate? Yes, sir. Yes? Okay, so is it then differential or selective? I heard both. Differential or selective? No. Yes, so it is differential. So think about like, it may be hard for you guys to understand this now, but I want you to think about conditions in which bacterium are not able to grow. If there's a high pH, there's high salinity, right? In the case of the MSA plate, it has a high, salinity indeed, osmolarity 7.5% sodium chloride, that's high. So that would be a more selective trait. Mannitol is a sugar, right, a metabolite. And if it's fermented, then actually the bacterium, if it's metabolized, the fermentation process will exhibit a change in color from phenol red to yellow, essentially. So that the two of them together, or at least mannitol itself, is differential. And then phenol red is going to be able to elicit that differential trait. So if it's red, or if, i.e., it doesn't metabolize mannitol, or if it's yellow, it does indeed. So think about those traits, and hopefully that'll make sense in due time. Does that make sense, folks? See, some of you are on your phones. Remember lab performance, folks. So now, are there any follow-up questions? Maybe anything that you want me to clarify? No? Yes. Great question. So it would technically be the one on the left. Reason being, You have actually two options, folks. Unless they're already separated, I don't believe they are. So we'll do exercises 1, 3, and 5 in your groups as before. But to make it easier, I would recommend doing 3 sections, so that way you serve one as a control, and that way you should be able to more directly see a change in color. The benefit of not just having 2, but 3 sections will also help you ensure that you have a control that isn't contaminated in your same plate, but also if there is a difference in hue, like as you could probably see here, it's more red here and more faintish pink on this side. So that will give you an idea as to, okay, there may be a differential, like there may be a degree, a lesser degree in which Staphylococcus epidermidis is able to ferment mannitol. So for exercise 1, remember all your plates need to be labeled, right? Exercise, group, practical, exercise, practical group exercise, and your different quadrants could be separated on the end for the different bacteria. Right. And this is just an example, exercise 1. And you would do the same steps as you've done from before, follow aseptic technique. Just make sure that you are able to take the bacterium as shown from Landman. So I'm just going to show an example. Let's say this is exercise 1. Group 2, group 1, and then you put the date. Let's say get this 25. So and then essentially the idea is that you want to put like your control on each edge. Like each edge should have a different mention of your bacteria, Staphylococcus epidermidis, and then E. coli. And then you're going to do the same for your other plates as well. But they have other sections with other bacterium. So just bear in mind, folks, that if you look at your plates, that it should be detailed, but you have on your red trays 5 different tubes. So there'll be, I believe, 3 or 4 with bacterium, and 1 is serving as a control. Actually, 1 is serving as your blank for the Spec 20 read. So I just want you for now to focus on plating your bacterium. And if you need help for me to separate it for you, I can, but like to separate the slides. But essentially, use the sterile cotton swabs that you have on your bench tops, and you should be able to, for exercises 1 and 3, split up and use the same bacterium, Staphylococcus epidermidis, E. coli, and an uninoculated, so like your negative control section. So just split both of those reddish looking plates into 3 sections, and then label them accordingly, and feel free to get started when you're ready. I'll give you until around 10 to like finish this because it's relatively simple. Remember, when you do your swabs, try to distinguish. You can make them longer, like or thinner, but try to make sure that you don't cross boundaries, right? Because you want to make sure that you're not touching and thus not cross-contaminating one another. I can also go into the presentation if that helps. Oh, yes. Well, for exercises 1, 3, and 5, you're going to be using the, well, you have the solid agar. Sorry, you have, let me just show everyone. Okay, so, for the blood, so make sure you have that now. That's for making the you have solid broth. Some of you have the Staphylococcus epidermis and some for subtilis. I've also seen some with coli liquid. So, oh, you have those. So essentially, use the liquid broth cultures for your swabs. Essentially, it'll make it easier, and that way you don't have to ask for saline solution, because I don't believe we have apart from the water there. So there is autoclaved water, but I would say for now follow aseptic technique. Remember that you've essentially flamed your tubes accordingly, and then we could go straight ahead. Remember, just stay within your section. And then there's other plates for the viable count. Which will be looking at for the Well, when you're doing this, remember you suspend your bacteria. Make sure that you are taking a spot inoculation with the cotton swab and then spreading across your section of interest. Make sure that you have 3. When it's saying spot, it's like you're dabbing in 1 drop into your liquid bacterium, so your liquid broth, and then you're essentially what do you call it? Like essentially pressing onto the edge to remove any excess liquid, and then you are spot inoculating as shown with the blue ink, so or the blue marker. So like you're spreading accordingly, because if you just do a spot, then you're just gonna see like you may either see just a dot, or you may not clearly see your results accordingly, because this is what you want to be able to see, right? You want to see like a spread, a smear of bacterium. Someone's Bunsen burner on too high. Remember to keep it low, especially if you're not using it high. Everything okay? So let's have a look here, answering this discussion question. I know some of you have done it in your lab notebook, and I'm very happy. But just to summarize this one, you have Staphylococcus epidermidis and Staphylococcus aureus. Remember, these are from the same, I don't want to say genus. They have very similar characteristics in morphology, right? And being gram-positive bacteria. And something interesting here is that between the 2 species, you can notice that there's a differential characteristic. One is mannitol, able to metabolize mannitol, the aureus, and epidermidis is actually unable to essentially metabolize mannitol. So these things actually have functional relevance because, for example, if you wanted to do the same example of transformation from a few weeks ago, but doing it with a Staphylococcus, either epidermidis or aureus, if instead of having arabinose, you had mannitol, which is a salt, then you should be able to know which one would grow and which one would not, right? So likely based on what you see there, would it be Staphylococcus aureus or epidermidis that would grow in an LB mannitol / ampicillin plate? Aureus, I heard aureus. Aureus, yeah? Great job. So think about that. These types of questions like extend from just not only from the themes and topics we've discussed from the discussion here, but also beyond them, and something that you want to think about for upcoming weeks. So once again, and by the way, I've seen many of you are done exercise 1. Anyone not done exercise 1 or 3 as of yet? Let me know. Okay, so briefly then, if you look at the quadrants here, and by the way, you want to think about this for your lab exam. It's coming up soon, so you want to think about different details and connecting the different concepts thus far. Biosalts, right, compare osmolarity. So think of that as a selective trait. Neutral red is differential because essentially following metabolism with lactose, there's going to be a change in color. pH is going to decrease, and accordingly, you're going to see a change in color from neutral red to a more yellow hinge, actually a darker red hinge, which is what you can see with the crystal violet coupled here. So crystal violet, differential, neutral red, differential, bile salts are selective. So think about these different traits and how, generally speaking, they influence your results based on the media that has been prepped for you. Let's go to the DNase, and then we can go to exercise 4. DNase, anyone recall what DNase is? Does anyone know the full name? Say it out loud if you can. Right? Yes, yes, yes. So that is correct. Deoxyribonuclease, and it is an enzyme that actually I want to ask, where does it break down DNA? So it breaks down DNA that is generally double-stranded, right? But based on what you know from probably Bio B12, does it cut in between? Does it cut at the edges? Does it make staggered ends or blunted ends? What do you guys think from DNA? Because expect these types of technical things. Repeat it again. Okay. Tunic? Is that what you're saying? A fork? Like during DNA replication? So it does that. There is a small component of it there, indeed, actually. But that would be technically the helicase. But nonetheless, the reason I bring this up is because, like the catalase test, like the beta-lactamase test, this DNA test is going to be selective. So you're going to try to essentially see, based on now simple streaks, like one-line streaks, what the results should be for your inoculations. Here's a good example. So in a week's time, you have, once again, the 3 sections like before, with the control, the subtilis now, and E. coli. But now the idea is that you should be able to see the results of, like an example here for Staphylococcus aureus, positive in yellow, if I'm not mistaken, and then positive also in pink with Saratoria or Saratia marcinus, and then the DNase negative bacterium Staphylococcus epidermidis. And so these different traits that we're learning about, we've seen mannitol positive for aureus, mannitol negative for, or actually doesn't metabolize mannitol for Staphylococcus epidermidis. Now we know that it also doesn't have DNase. So think about these different concepts, especially when it comes to your lab exam. Any questions? I know some of you already started the DNase plate. How many of you have completed it? Raise your hand. Your group is waiting, almost done? Okay, and your group is almost done. Okay, so let's, in the meantime, Keep it up, you guys. Keep it up. So in the meantime, for those that are waiting, and even from the groups, if you have peers of your group members idle or paying attention in the meantime, you can actually get started with Jelani's test. Let me just give you some pointers. And by the way, Cedric, you can see that, right? Okay, perfect. So you have also cubes from last week here. Don't concentrate on them as of yet. I just want you to pay attention to the following for the dividing test, because it's very important that you make it correctly. Think about the motility test, right, from week 2. You have 3 sets for each group. 1 for each treatment plus a control. The subtilis and the coli, just like for the DNase plate. Now, test plate. Now, the thing is, remember for your inoculating group, you're using the solid agar, right? So bear in mind, you're using the solid agar tubes. And once you've taken, okay, think about a nail thing. About a centimeter long into the bacterium, not into the agar, but like touching into the surface of your NAS plants, your bacterium, your solid agar plants. Your goal is to essentially place a pathway into each of those different tubes. Please bear in mind the septic technique. You've labeled your tube, right? So the 3 different ones for control. If you don't have to touch, label, and then the other tube that you're actually going to tap into pathway to each of those main. Do it just once, and that should suffice. Let me know if you guys have questions. Otherwise, if you have the groups, you've almost done exercise 3. Done? Okay. In the meantime, you guys can get ready. Make sure you label your tubes. Continue aseptic technique. And you just need to open the 2 experimental tubes for B. subtilis and E. coli. Yep, you're just poking in. No problem. I'll give you about 5 minutes to do this one because it's not difficult, trust me. And if you need help, ask your other group members. Try to make use of your whole team. That way, inshallah, it'll make things easier for you. You don't have to worry about like doing it all from one person's hands because it's very simple and it'll give you the technical skill to be able to inoculate the different bacterium following the aseptic technique. If your group is done exercise 1, 3, and 5, you could tape them and invert them, having the agar on top into the top left incubator. Please make sure that you place them taped up, and so that way it'll make things easier. Include the group number as well, so that way it makes it easier to discern afterwards. So in the meantime, have your group members split up. You have likely 3 to 4 members, 4 people per group. So 3 of them could deal with exercise 4, and the last person could focus on placing back the plates into the incubator on the top left side. Because the control is also no inoculation, pretty much the other group member could focus on looking at the other results. They can bring the other tubes from week 8 into their benches as well, including the plates for viable counts. Someone's Bunsen was on too high. Please ensure that you lower it accordingly so it's not on too high. That would be a B.
For the exercise 7, we need a TSP boat to make it liquid. Where is the group?
Great question. So let me say, essentially, don't worry about exercise 2, 6, and 7 for today. 7 is going to come up next week, so we won't have to actually do it next week.
If we are done with the exercise 2, we are good for today.
There are a few other things.
Yes, we're gonna do the week 8 now.
Yeah, perfect. If your group is done, by the way, are all groups done exercise 4? Please raise your hand. Group 3 is done. Group 5 or group 6 is done. Group 4 is done. Group 1 is done. 2 and 5. We'll wait for 2 and 5 for about like 2 more minutes. Is that okay? Is that okay for you guys, or you need more time? Okay. In the meantime, groups can start looking at their results. Actually, let me go back. No, so week 10 is actually not just a review. There's still a few things. You'll pretty much be looking at your results from this week and essentially doing very simple experiments.
It's not only review. Oh my God, you just make my house.
Well, it's simple. It's not. It won't be. It'll be hopefully easier than. Are you done? Yes? Okay. So technically, folks, if all groups are done, we're gonna place them into that middle rack, and then one in the middle bench. So see that pale, whitish looking one beside the autoclaved water. Place your tubes there in order. For group 3, I believe you would be a bit more down. So just try to make space, orient yourself so that way once next week comes in, inshallah, you'll be able to organize and see, oh, this is my group. And this is like, okay, where I'm going to be putting all groups are labeled, right? They're all labeled. Oh, sorry. So when your group is ready, you can place your plates as well. The plates that I talked about earlier. We're gonna place them all into the incubator, including the one for today, for the July test. So that way we can see the results. So don't worry about exercise 7, exercise 2, exercise 6. We're going to talk about them. And so just bear in mind that you're able to complete exercise 2 from last week. So bear in mind the week 8 results. From last week, find the last 2 in your manual. And you should be able to see your results in putting on the floor. Some of you may be wondering, I am putting the Jazakumullahu test tubes actually into the incubator. It's recommended by the lab tech, so that way results will be done more yes, faster. And essentially, we can see you can see it a little bit. Actually, there's 2 options, 25 for longer. That's how it is. Does everyone, finally, just to confirm, everyone's done exercise 1, 3, 4, and 5?
Yes.
Yes. Amazing. So, let me finish week 9 material. And you may be wondering, what happened to, week 7 material? Sorry, exercise 7 or exercise 2 or 6. Let's talk about them. So, we finished the DNase test. I wanted to also talk about this. So bear in mind, you're all paying attention now. So for the DNase test, even if the results appear, sorry guys. Okay, just have a look here. I want you to pay attention to the results. So after 24 hours at 37 degrees or 25 degrees, you're going to essentially see a change, viscosity of your media, or in this case, of the gelatin found within the media itself. And something to bear in mind is that if bacteria have gelatinase, right, an enzyme that will break down gelatin, then the idea is that you're actually going to see, and I know it's interesting. So the lab tech told me, yeah, actually, just don't worry about some other things. But in the meantime, think about the idea that if gelatin, like in different um, agarose, sucrose, gel media, like if essentially you have gelatinase eating up that gelatin, then it's going to become more liquefied. So think about like some of you have seen from our plates before, some of them that are newly incubated or fresh, very fresh media actually are still like solidifying, right? And you probably have seen some of you liquid media on the plates, right? So the idea is that after 24 hours, if there is the presence, or actually, if you essentially find your tubes liquefied, then that exhibits that gelatinase is present, and that's how essentially, or one avenue. Let's see if there's any extra things. Yeah. So essentially, if I want to say this in the best way, think about your FLRs. If you observe viscous liquid media in your tubes, then that exhibits, or you're observing, essentially bacterium that have gelatinase, right? So thereafter, in brackets, essentially, if you observe liquid media, then the gelatin found in that media would have been hydrolyzed or dissolved, and thus you have it presented as liquid shown in tube A. So, and then in contrast to tube B, right? So you could do like a very uh, small tilt, and likely you wouldn't really see even a larger tilt, a great difference, right? The gelatin in the media, or the media is still solid essentially. So think about it like that. If it's positive versus negative. And that's something that we hope to see next week. So think about those results. If you had a control tube, um, actually, this is a good question. So look at tube A. If tube A was your control, would that be good or bad? Bad. Why? Why would it be bad? Would it be bad? Yes, right? So think about it like that. If it's like your plates, right? If your plates are liquid, how are you gonna do spread plating, streak plating, right? That would give you a big concern. And in similar regard, your control should be essentially without any bacterium, right? So if there was bacterium and it indeed was gelatinase positive, then that's where your experiment may be problematic. For both your experimental and control conditions. So bear that in mind. If your control doesn't appear good, then you may have to question the results of your other conditions, including like tube B, tube C, etc. So we went over DNase. Oh yeah, I didn't talk about this, but methylene green or methyl green is essentially your selective dye, which many of you will see, right? Like if it's essentially a dye or a color, like neutral red, crystal violet, methylene blue, methyl green. These are all like selective, sorry, differential markers. Whereas like when you have specific conditions of like high pH, or sorry, low pH, or high concentrations of NaCl, or salt essentially, even metabolites, those usually, sorry, outside of metabolites, but those more extreme conditions exhibit a more selective trait. And then when you have metabolites, those are generally more differential because you will have byproducts that essentially exhibit or depict whether or not that bacteria not only survives, but has specific metabolic traits. So it could maybe metabolize sucrose, but not lactose, or it could metabolize pectin, but not gelatin, etc. Any questions, folks? No questions? So now let's have a look at biofilms. So this was probably your question, right? What's happening to biofilms? So the lab tech that's actually going to help us. He's going to take care of, he's going to give us the results for lab 2. So week 2 protocol essentially is going to be done next week. So essentially you don't have to do anything for today, but the idea is that he's going to give you the tubes. So there'll likely be 6 of them from Staphylococcus epidermidis, but the idea is that they're going to be see-through or like transparent. So think about like Staphylococcus epidermidis is relatively pale. And so the idea is that you're going to see a tube that's going to be like the one in the middle, but very faint, right? So then what you're going to do is use crystal violet, which is going to be like a really nice full circle for many of you with the gram stain, as you recall, to essentially just let it incubate for about five minutes, if I'm not mistaken, if not even less, and then essentially let it rinse essentially the crystal violet off and place it into the whiskadine, and then thereafter let it dry, and you'll see the differences in colors. And that pretty much helps you to understand and appreciate the roles of biofilms because they have unique traits. If you have seen from your lab manual, essentially biofilms make up extracellular matrix, but also bacteria themselves. So think of it like a hub or a grid of multiple bacteria that are able to ferment and ferment and metabolize within a given surface area. And so essentially they have sugars, metabolites, waste products, and many bacteria, which is why you can actually see directly without using a microscope the clear, dark purple pigment, because these are, after all, are something to keep in mind, gram-positive bacteria. So think about that. Think about it in terms of the results, and that's what pretty much you would see for exercise 7. So that'll be something to think about for next week. Any questions? Biofilms is a high yield topic to think about. So think about it a bit, understand the different concepts that we're talking about for your lab exam. Now let's go down. Snyder test. So essentially these ones, exercise 2 and 6 are virtual, meaning that we're talking about them here today. But the idea is that if you have a look here at for the Snyder test, it's actually really interesting because this connects to those that are interested in dentistry or even like other like healthcare related fields outside of medicine or even in microbiology. The idea is that you have, like before, different components making up your media that are shown here. So essentially metabolites, color, almost crystal green, and then you have pH. So I'm going to ask you guys, is pH a selective or differential component of the Snyder test? It's selective. How many of you think it's differential? Raise your hand. I aim to be a bit tricky, but okay. How many of you think it's selective? Raise your hand. Okay. How many of you think it's differential? Okay. Okay, let me go down into the details so you understand where I'm going with this. Okay, you have 3 highlighted ingredients, right? All the other ones are generally needed, right? Even NaCl, if it's not high concentrations, it shouldn't be selective. So keep that in mind. You have essentially, as can be seen here,
interesting notes indeed, actually. But the idea is that you have your bacteria. That could be lactobacilli, or what we even recall B. subtilis, right? Or even, yeah, essentially lactobacilli, essentially those that ferment lactose, and these are usually rod-shaped bacteria. But the idea is that following their growth and their metabolism of substrate, which is in this case glucose, which is differential, you should thereafter see a change in color with bromocresol green, right? So think about it like this. If it's a bit confusing, you have two things. Think about like a chemical reaction. You have your bacteria. They're on the reactant side. Are going to react with a metabolite. Okay, bacteria has metabolite. That could be sucrose. It could be a sugar. It could be nucleic acid potentially. It could be a protein, polysaccharide, etc. Or like a lipid. And then essentially following that, you have a dye in the middle. It's not a catalyst. The idea is that it's present in the solution anytime. Now, with that reaction of the bacterium and the metabolite is fermentation, right? And so pH is generally speaking going down for most bacteria. As it goes down, that actually changes the color of your dye. And so it goes from a dark forest green look essentially, or dye, or hue, to a more yellow-green hue, which is why you essentially see a change in color. That is differential, right? Because bacteria, whether or not they're like, if you see these two bacteria, sorry, these two, they both can have bacteria, right? They can be both surviving. But the difference, what's making it differential, is the fact that this one essentially metabolizes a given ingredient or like a biomolecule, that is sucrose, dextrose or glucose, which is a saccharide, and then in contrast, this one doesn't. So essentially, you'll see different colors. You'll see forest green, light green, maybe turquoise if it's contamination or things of that nature. So you'll see a spectrum. That's more differential. When it's selective, as in the case of pH, then that's where essentially you have a change in color. Let me just give you context. The pH is essentially indicative of the sensitivity of your bacteria, right? So if, let's say, how do I say this? If your bacteria can grow at, let's say, acidic levels, and maybe it's a basophile, right, or a mesophile, then what you would expect is that you wouldn't not only see change in color, but you would likely not even be able to, let's say, regrow those bacteria outside of that tube. So the idea is that pH, generally speaking, is a selective trait. So bear that in mind, folks. Bromocresol green is differential, glucose and dextrose are differential as well. Any questions? Yes. Yes, or even more particular, if it's selective, it's as if like the trait itself would essentially show if it either survives or it doesn't. So the gram stain, think about your stains of the past. Gram stain is differential, right? Because you have crystal violet and safranin, right, following the two steps and the wash in between with the decolorizer, you are essentially having two different colors. You have purple and pink, gram positive, gram negative. That's differential. If it was selective, like essentially hypothetically, if before you started an experiment and you, let's say, used ethanol and sprayed ethanol on your bench and then did a spot inoculation on a plate, then you would likely only get bacteria that essentially are resistant to ethanol, right? So that connects us back to our germicides exercise from the past. But the idea is that if you have bacteria that can grow in certain conditions, and that condition is present in your media, then that condition itself is selective. Hopefully that makes sense. If it's able to grow, but shows the differences in colors, then generally speaking, so that way you can characterize or identify them or distinguish them. That's where the differential trait comes in. So like in this case, I actually want you guys to do this for your, if you haven't done it for your post lab, I want you to do this question because I remember it's actually a pretty nice one. And I want you to do it. So that way you understand the concepts. Exercise 6, blood agar. So this is an interesting one, and one that you should keep a close eye on. So you could see that there are 3 different types. Actually, if you saw from your lab manual, everyone saw like the different toxins, the different like the hemotoxins, the pretty much the different categories that they have. It's very important that you understand this for different reasons, but being able to understand what I'm talking about today will also help. First off, so if you recall, I don't want to waste time, but essentially, there are 3 main categories: alpha, beta, gamma, right? And so from left to right, as you can see from the slide, beta is going to lead to complete hemolysis, meaning that like you're going to be rupturing the cells, the RBCs, the red blood cells, drastically. And that essentially leads to the secretion of hemolysins, wherein you see a direct change in color that's shown in yellow. That indicates a differential nature, right, of your media. So I want you to keep that detail in mind because if you compare beta to alpha and then gamma, they are actually in a spectrum, right, from complete to partial and to null respective hemolysis effects. So just bear that in mind. Beta hemolysis leads to complete hemolysis, alpha hemolysis leads to partial, and then gamma leads to none. So, but the idea is that you want to be able to understand these concepts because when you deal with different types of Staphylococcus and Streptococcus species of bacteria, then you'll notice that they have different, paths of hemolysis, and they have different, they function in different classes. So bear that in mind. And that actually has great clinical importance or relevance because if you think about the conditions here and the different types of bacteria, and obviously no one wants to be infected with bacteria, but think about the consequences if a specific species, not just of Staphylococcus or Streptococcus, but if it was Streptococcus pneumoniae or Staphylococcus aureus, what the different consequences would be. If you think of Streptococcus pyogenes, this is one that you want to keep in mind, as it's a unique species as well. Does anyone have questions as to why at least this is a differential and not a selective plate? Think about once again your results show differences in color. That's a differential trait, right? Just like morphologies like we've seen in the past, or even like, like we had of cross-contamination with different like size or shape or colored colonies that you've seen from your plates, or even from your stains, right, from weeks 1 to 2. So think about that. If it's selective, then you would have specific conditions that would make your bacteria grow. That should make sense. If not, yeah, so essentially think about those different traits. They're also shown here, greenish yellow colorations. Fun fact, these are class contaminant level 2, if not 3 bacteria, so you have to be very careful. And that's why we're actually not doing it as well. Well, to consider your safety, of course, indeed. Now that we're done though, actually before we're done, any questions for week 9 material, weeks exercises 1 to 7? Are you guys bored? Don't be bored. You're almost done. You're almost done C17. So okay, let's go to the fun stuff. Looking back into the past of week 8 material into your environmentally conditioned tubes. So has every group grabbed their tubes, by the way, just to confirm? You don't have to do a presentation. You don't have to, but I like to ask some questions, right? Because I haven't been able to give you guys questions as much. So we'll spend a bit of time to do so. Let me just see. Okay, let's ask those questions for now. In fact, actually, has everyone gotten to look, has everyone gotten to see their tubes for week 8? Like all the different ones? Hopefully you didn't grab more than one set of tubes. Just to confirm, every group has grabbed their tubes from last week, yes or no? So I did 2 different sets of arrangements. Yes. So the plates are for the TDT exercise 4 from last week. Every group should have their own, as well as a group with their antimicrobial susceptibility test plates. You could take photos and return them back to the middle bench. Don't dispose of those plates. But for the TDT, what you can do is actually, I don't know if you have plates. All plates are comfortable. Okay, that's okay. That's okay. So for let me see. Let me. Okay, what's your questions? That's what we're gonna do. But before we do that, I want us to do 2 things. One is so I want to make sure we have time. Okay, so there's 2 things that we need to do before we go back to the slides, and they are to look at the TDT results, to count the number of colonies, recall 30 to 300. If you have less than 30, that's okay, but account for the dilution factor, and I want for your group to place it here, potentially. So place your final readings. So for example, let's say you have 17, maybe not 17, 124 colonies and a 10 to the minus 6. Actually, I want to see what we, for your group, do you guys have 10 to the, what do you guys see for your colonies? Where do you see them countable? Are they like in 10 to the minus 5? What is that? 10 to the minus 5 and 6. Okay, so I want you to essentially count them. If you have 2, all of you only have 2, by the way, out of the 6 per group or per condition. Do you only have 2 plates that are usable or countable? Yeah. Only 2? Only 2? Only 2? Okay, so we're gonna make so place in your final value So, sorry, let me clarify. You have plate A and plate B essentially, that you're going to be writing down the final PFUs for, and then calculate the final as a mean of the two of them. So that's going to be, so don't worry, in terms of your groups, you just only need like 2 people to work on that. But in the meantime, your other group members could focus on the spec 20. Yeah. It depends because if you, let's say I 10 to the minus 6 and 10 to the minus 7, I didn't include it there. That's why I just placed A and B to make it simple. But you're going to put the final, folks, let's pay attention, please. You're going to put in the final sorry, did I say PFU? CFU, viable counts for each plate that is countable for your condition. So if that plate A, let's say it's 1.3 times 10 to the power of 7 CFUs per mil, you're placing that there. Then you do the same for your plate B, and then you calculate the mean for those 2 plates. Every condition is going to do that for the TDT readings, and that's going to help us actually record the values to determine the TDT and D values for exercise 4 for last week. In the meantime, because that only takes about 2 to 3 people to work on, the other remaining group members could focus on taking the spec 20 reads from the blank. So make sure you have in blank the spec 20 with the tube that is shown without a label. Yeah. So like one that's see-through, like very transparent, like the one that Laiba's showing. And you're gonna essentially blank the spec 20, and then I want you to record the and how you're gonna do that is actually just put half. Let's say you have a pipette, all of you have 2 pipettes. Use one for the blanks, one for your experimental, pretty much of your osmolarity conditions. And the idea is that, let's say you want to know how I'm going to essentially do this display. You just pour it in actually directly. Just be steady that you don't like spill it off. But if you need to, pour it gently and then place it up to at least halfway for each cuvette. And then once it's blank, place it inside and record your observations just for osmolarity. You won't have to do it for the other ones. But after that, or following that, we'll come back as a group to look at the results for effects, the temperature, and pH, as well as osmolarity for visual qualitative observation. Any questions, folks? No questions? Is that clear? Yes. So we had for osmolarity 4, if I'm not mistaken. Was it 4 or 5? 4? We have 4 conditions, but I don't recall because I had different groups before. That's why I don't know if I split you into 5 or 4 for that one. 4. So actually, what you could do is, by the way, make sure to put in scientific notation. Have it like 3.5 times 10 to the power of 7, 0.3 times 10 to the power of 7. But keep going, you guys. Oh, use permanent. Yeah, okay, don't worry. Let me just recall something. Yeah, so that makes sense, I hope. Questions, folks? Have your group members split up, focusing on spec 20 reads and for the TDT results for observations. So if you need help blanking or if you need help recording your observations for osmolarity, let me know. And you could be able to do this in the next. I'll give you 30 minutes. The sooner, if you're done sooner, then that's even better. Yes. So, okay, that's a great point. I spoke to the lab tech last week, and he actually didn't want us to do the extra one. So that's why it's okay if you don't have it. We're gonna look at them all as we go. So don't worry. Yes. Oh. Any questions, folks? If anyone has to like go outside briefly, use the washroom, let me know. You can, of course. As Stephanie did, you guys can ask me and I'll be more than happy to respond accordingly. Any questions? Try to focus on splitting up, recording the values. Okay, this is good. This is good. I think you're good. Any questions?
I guess missing time. They're there. No, we have to make sure that any further. You're writing it, not mine. Why did you? That's not my writing.
Group 3, are you guys missing osmolarity readings?
So we write our fraction wrong. Yeah, that was
If anyone's missing tubes, feel free to let us know.
No, we have that.
That's good. Yeah.
It was the control.
Yeah. I think we have extra, so maybe just an extra. Maybe that's
That's ours? Yeah. Yeah, okay, that was the registry. Yeah. Yes.
Make sure we suspend your tube.
That's not mine. Yeah, me too. I don't know what these things are though.
Okay, I'm just gonna also come back. I'll use the washroom. Try to finish pretty much the calculation for the viable counts for GDT meeting and and then also same for the environmental treatment conditions, temperature, salinity, osmolarity. You guys can complete those in the next time.
Sorry, just 1 second. Oh, the mean. All similarities, we will need to write down, right? Yeah. Okay, so we would break it down. This is like one, six. Serious? Oh, I do.
So let's come to the fiber here. No class group, right? So I see we have our results. No, actually, so the results here are okay for 65 degrees, but something looks off for 15 degrees. Something wrong? Either with, like, actually, let me ask, what do you guys see? Like, what do you see as you increase in temperature? Growth should increase or decrease? It should decrease, right? It should decrease. Now, if you go up in time, should it increase or decrease? The viable counts. Decrease? Okay, how about this? I'll wait for you guys to finish so we'll talk about TDT results shortly. How are we doing for the spec reads? You guys are done? Is anyone done or midway into the spec 20 reads? Yeah.
So do we just pour it in and then clean it and then pour it in?
You could do it, yeah, pretty much 1.1.
Yeah.
But the question I have is what is the really So let me know how it goes for you guys. The idea is that you first want to blank your tubes. Okay. Does anyone need help starting off their spec 20s? You're almost done? Perfect, okay. That's good. Classify that directly, place it inside. If you do that, Members, I want you to know clearly recording from UTSC. You want to make sure you're able to record them lively. By the way, folks, please make sure that before you dump your tubes into the Western diet, that you've been able to at least visually compare the differences in the different bacteria, the cause of concentration. Yeah. Folks, I need you to all send me your readings for the Spec 20 results. So keep in mind, there's the qualitative observations, right? So that we could do as a group. We could, and you probably already had a scale like from 0 to 3 or 0 to 5 or 1 to 3. We'll talk about that as a group, but I want you to send me those observations, the qualitative ones for the osmolarity and for your quantitative Spec 20 absorbance readings. That way we can aggregate all this data in and then hopefully include this into your post lab as well. Are we good to complete this in the next 5 minutes? Yeah, next 5 minutes, okay. I have a question for group 1.
But the other half possibly absorb it. And I sort of, we always use a blue that better is like funny and something. Well, there's probably enough for it to be like, so. It's almost like, wow, how many artists I just love it.
Blind still, by the way? Is it okay if you could use it?
Because I'm not gonna do
There is. I use it when I work with one thing. So actually, I just have stuff, samples to
Yes, there is a formula. So, folks, we'll talk about. Oh, sorry, you want to say something? Negative absorbance, not negative transmittance.
So the negative along, and so it results in a negative.
Okay, let me explain.
Maybe I didn't know. Yeah. Basically, I use this one. Here's another one. Yeah.
I'll talk about this. I think now's an ideal time to talk about something that is actually very important to understand for your lab exam. So it's connects back to lecture material, and some of you may be wondering because we have actually started with transmittance, rather than absorbance for your spec 20 needs. And so there's a few formulas. One is actually shown earlier, I believe, Daniela. That one. There is another one that you would normally get more results from. I'm just going to show these formulas. This is 1 centimeter, the length of your cuvettes, the concentration of the solute, and absorbability coefficient. This is like how much, or essentially how readily accessible or absorbable your material is. So it's the absorbability. I don't know if it's changed from the time I took this course, because normally you do learn this from like first 2, maybe first 3 weeks of lecture material. I don't know if you haven't heard about this. If you haven't seen this formula at all, or anything talked about in class about like transmittance versus absorbance, no. Okay, so let me help you guys out, because this is very important. If your values from transmittance are negative, let's say negative 5.3, negative 2.4, negative 7.5. So like think about it like this. Transmittance is inversely related to absorbance, right? So if you have a cuvette like this, and it's absorbing solution, like in the case of like how turbid your bacteria are in your solution, if it's absorbing, because these are at wavelengths of 550 nanometers. So the idea is that whatever is being absorbed is going to be kept inside, right? It's going to be essentially subtracted from what you see from the spec 20. And so whatever passes through is going to be transmitted. Whatever is absorbed is reserved as absorbance. And even though you're correcting these values and doing some things on the spec 20, the idea is that the absorbance values give you positive reading. Putting a plus sign to give that indication. So the idea is that if it has, like the more it absorbs, the higher the absorbability, actually the higher the concentration, because this is generally controlled, the higher the concentration of your bacteria, the more turbid it is, the higher your value of absorbance is. And because it absorbs more, that means that it would transmit less through the cuvette, through your glass tube, the smaller one that goes through your spec 20. So in short, the higher your absorbance, the lower your transmittance. How does that connect to this? Well, consider the same thing. If your values are negative but are going up for transmittance, right? So let's say in terms of order, like I'm going to organize this on the left-hand side. You have negative 2.4, negative 5.3, and negative 7.5. These are going down in transmittance, meaning, like for cuvette 1, well, I'm saying 1, 2, and 3, because they go down in transmittance, I need less and less is passing through the cuvette, then it's increasing in absorbance. Meaning, your first tube is going to be the more turbid one, and then, or the more concentrated one, i.e. the one that has more bacteria, and then it goes down for tubes 1, 2, and 3. So essentially, the idea is, if your values are more closer to like 0, or they're less negative, then they're the more turbid readings. And that's going to help at least for qualitative observations. So don't worry if because this may be the case for half of our group. I won't expect you guys to actually pull it in all together as of yet, but you do want to record these values in your post lab. So that way you know those values and you should be able to thus deduce, okay, which one is more turbid For example, if I had E. coli, Staphylococcus epidermidis, and B. subtilis, then I know that, let's say, if B. subtilis is number 3, Staphylococcus epidermidis is 2, and E. coli is 1, then I know that E. coli, let's say at a saline condition of 3% NaCl, I know that essentially I can tell that actually I'll write it down so you guys can see what it means. Let's say this is E. coli. This is Staphylococcus epidermidis. And this is B. subtilis. This is an example, right? This doesn't have to be your result, but I'm just saying it as an example. That if these are your results, then you should know, even for like understanding these inferences for your just to understand the material more critically, you should thereafter know that let's say this is at 5% NaCl. I know that E. coli goes the highest and B. subtilis grows the least or lowest. And that reflects, now you want to take it a step further because based on those findings, be it for temperature, be it for salinity, be it for pH, this helps you categorize what the bacteria are. Just like how we did for the catalase test, you saw some bubbles. What does that indicate? The level of catalase present in your bacteria. So in this regard, if it grows highest, let's say your E. coli, at a higher range across different, concentrations of NaCl, then that depicts osmolar tolerance. And even though like it is saying that it's growing the highest here, you would have to see this across different concentrations. So bear that in mind. So you would have to see this at 8%, 11%, and at 2%. But if the same, if like the trend continues as such, then the idea is that there is osmolar tolerance for E. coli, right? At 5%, at 8%, or maybe at 11%. The higher, essentially, the concentration of your solutes, the more osmolar tolerant your bacteria are if they have essentially at those concentrations. Does that make sense? I heard no. Is that anyone having problems on this side? Understanding the material? We won't have to present, like what I do want us to do before we end off today actually, is two things. Thermal death time. I want you guys to understand this because it's very, very important. You're going to be drawing it out in your lab note. So bear in mind your results across qualitative and quantitative observations for last week's week 2 environmental conditions. All of those values you need to have in your group in your post lab. Bear in mind you have a table detailed with captions and units and everything that we've talked about thus far. For your TDT results, this is something to point out. You are going to do this in your lab notebook. You can use semi-log paper or you can do this manually, and I don't mind. But bear in mind that you follow the log scale. So, for example, let's say, and this is how I would do it. You see, everything here is around 10 to the power of 7, right? So really, you're going to go up from 10 to the power of 7 to 10 to the power of 8. So 10 to the power of 7, and this is 1 times 10 to the power of 7, all the way to 1 times 10 to the power of 8. Meaning that your increments, which are going to go slowly, like they're going to go rapidly up at slower, larger increments, like this. Maybe hard to see, but like I'm doing it as a sketch. So maybe technically 1 times 10 to the power of 8 is here. But the idea is that you would actually plot this down. So at 50, so okay, let's see at time, because you're doing this not just for, by the way,
Actually, let me see. Did you guys do this for? If you okay, I would at a minimum recommend doing this for a microplate reader because you have it more definitive across time. But if like to keep things consistent, but in either case, let's say this was a microplate data. You have it at 0 minutes, and it's okay if you're outside. But I would recommend for your lab exam, if you have a question like this, start out 0 here. But I was just making it easier so that you're not confused. The idea is that you have 0 minutes, 5 minutes, and then you're going to have 2 different labels for the different different temperatures. But ideally, what you should see is that, let's say, and you're doing the mean values, by the way, so you have that mean viable counts, units, CFU per mil, right? The idea is that if, let's say, I'm plotting this data, let me tell you what you should see, generally speaking. It would be, let's say, circle, just to represent an open circle for one condition. And then a greater jump. And this is just examples. But literally, even with these 2 different points, you can make a curve. And the idea is, and folks, I want you to pay attention because it's very important to understand. And I'll take time if you want me to, like, I want to go through it as effectively as possible. So just bear this in mind. You plot the data, be it the mean values that you see here, or on your microplate data, and you're going to plot them on semi-log paper like this, or you can draw it out as a sketch. And then the idea is that this is going to give you a thermal death time, right? The time that it takes for, ideally, we're going to shoot all of your bacteria to cease living. If you were to actually do the thermal death time, because often, like as you could see here, all of these are still live bacteria, right? They're in the millions, in fact, right? So they're not at 0. But if you were to extrapolate actually to essentially where it would reach the x-axis, that would give you your TDT values for at 50 degrees and at 65 degrees. And I want you guys to think about it. Which one do you think would go down quicker? 50 degrees or 65 degrees? 65, right? Now, it depends technically on your bacteria because some are thermophiles, some are, or mesophiles. So that's a point that's separate, but I want you guys to bear that in mind for recording the TDT. That's one component. So think about like your generation time assignment, right? You had semi-long paper, you plotted down the values, you denoted the values and made a curve, and then now you're extrapolating to determine the TDT, like your values accordingly. The other point though, and bear in mind that you have a label, right? 50 degrees, for example, 65 degrees here. You can use different shapes or colors, or just essentially different ways to identify or separate the curves. The other point is your D value, your decimal reduction value. Does anyone know what that reflects? I'm going to try to do this in another color. Because it's a different value, and so you want to know what it reflects. It's the time it takes for how much or how, like in terms of percentage, 90%? 90% of your bacteria to lyse or to pass away? 90%, right? So the reason why it's 90 or why this is relevant, in fact, is because it makes it even easier for you.
what I mean by this is that instead of looking at like the TDT readings, look at where you started off. Let's say, for example, let's say it's 3 times, or in this case, to make it reliable, let's say it's 8 times 10 to the power of 7. Let's say this is where I started off at my initial point. You're literally going to 8 times 10 to the 10 to the power of 6. Because essentially, by going down tenfold, you're essentially going down 90%. If there's trouble understanding that, let me know. But think about it like this: you're going down 10 times, going 10 times down, and so the portion that you have relative to 4 is 10% remaining. So it's like you're going down tenfold, and the amount that remains is at 10%. So essentially, hypothetically, if we kept going down this curve, if we kept going down all the way to 8 times 10 to the power of 6, then essentially, wherever that would lead you to, like that value is going to give you in minutes the time that the time it would take for your bacteria to reach 10% in growth, 10% in abundance, or essentially reduced by 90% in size population. So you're going down 90% or 10 times. 10 times that. And so the idea is that you want to record both the D values at 50 and 65 degrees. And I want you guys to predict this now. Do you think for like what I've shown here, do you think the D value would be larger for E. coli at 50 degrees or 65 degrees? Think about it. 50 or 65? 50. So bear that in mind. And that's important, right? Because if it's able to grow longer at 50 degrees, right? If it has a higher population, then it will take longer for it to reach 90%, like to reduce by 90% in size, or to reach 10% of its remaining population. So think of it like that. You need to record the TDT and the D values and use those values based on what you see from here or from the microplate reader to not only describe your results, but to interpret them and to actually compare them with what you would have expected for E. coli. Recall from generation time assignment. E. coli grows optimally around 25 degrees, 25 to even we'll say 37. And this connects us nice with the final set of slides because there's a range, right? As I talked about. And so the idea is that if it's beyond that optimal range, either too high or too low, there's going to be a decrease in that population's growth. And likewise, that would impact the generation time. So think about, connecting the dots from the past. But briefly, if you're increasing, the temperature beyond the optimal time, then that would thus lead to you seeing the reduction in that population, right? And so the further you increase that temperature, i.e. Hopefully that made sense because I didn't mention about the categories of these different conditions. But the idea is that bear in mind so bear in mind actually from the 4 different bacteria that you see here, they have different conditions, optimal environmental conditions that they grow under, right? So for E. coli, I mentioned it earlier, they have a more mesophile range where they're able to grow 25 to 37 degrees generally. It's a 20-minute generation time. Bacillus thermophilus, as you can see, the thermo indicates that it's able to grow at higher temps. So bear in mind, these different graphs can actually differ in range, but also in shape. Bear that in mind, folks, because it could go the other way around for a thermophile, right? Even though this may appear the case for a mesophile. And you can actually see similar trends, but expect that you may see unique trends and patterns for different bacteria. As you can see here, saprophiles live more in the colder regions. Think of the Arctic Ocean. Mesophiles like room temperature in our bodies. Thermophiles, so hyperthermophiles, those are the ones that are near hot pools, hot springs. Think about Taq polymerase, right? For those that recall from B-level bio courses, it's able to be active at 70 to 80 degrees. So you can imagine the bacteria that, synthesize the polymerase or like construct the polymerase are able to thus grow and live and replicate or like live at high temperatures. So bear those details in mind for temperature, and hopefully that'll give you an understanding as to how that would impact TDT and D values. If they grow at higher temps, bear that in mind. So let's say instead of E. coli, this was Sterothermophilus or Bacillus genus. You want to bear in mind that if it grows at higher temp, how would that then influence the TDT values? Would it be a problem for it to grow at either of those conditions, or would it even go up, right? So think about that. And then cryophilus is the opposite, more like a saprophyte. Cryo refers to like cold, so you want to consider that. And then for the other two conditions, let's talk about pH briefly. It's similar to temperature in its curve, but you do want to bear in mind that there's an optimal range, and that based on the different conditions, they are able to, and they likely will be hints on this for your lab exam, based on what Dr. Brent talks to you about it. For example, if she says like and by the way, I'm just coming up with examples, don't cite them, but the idea is that if you have, let's say, bacteria growing in your gut is an acidic environment. So you should likely assume then that if you were to grow, let's say, Helicobacter pylori, essentially a type of bacteria that H. pylori essentially is a bacteria that grows in your gut, actually in your stomach lining, when someone has like ulcers. The idea is that if you have this bacteria now outside of the body, from in vivo to in vitro, what would those results appear like? Like, what would you want to do to maintain its optimal growth? You would likely want to have it grow in acidic environment, right? So bear in mind, like the Snyder test, you would have a pH that's relatively low at an acidic level, right? So like below 7 for pH, so like 4, 4.5, maybe even in the case of the stomach, around 2 to 3 in pH. So think about how this connects. Microbiology connects to biology, connects to medicine, and connects to pharmacology, connects to many things that we've talked about thus far. And the last part that we could talk about is osmotic pressure or osmolarity. Yes. Okay. Yeah. We could try it on another spec 20. Don't worry about it for now. But we'll come back to that in due time. They can use it. You can remove it. You can also, if another group doesn't mind, we can move it to another spec 20 just to cross-reference because they should already be blank. But great question. Briefly though, osmotic pressure, right? Think about the osmolytes or the essentially, you can think about different gases, the different solutes that are present in your solvent, and essentially that builds up pressure. That also connects to human physiology because you have like lungs and like the different pressures that are influenced by CO2, O2, oxygen, etc. So the idea is low. Osmotic pressure influences the rate of osmosis of your in a fluid, and the more, let's say, NaCl or solute that you have, the more the increase of osmotic pressure you have in that solvent. And so in the case of like adding in now in your solvent bacteria, how do they react? Well, that's where you have the trends shown here. If you increase in NaCl concentration, some different bacteria are able to grow better than others, right? Halotolerant bacteria are able to grow and then actually grow at very low concentrations and then actually decrease thereafter. So they're very sensitive, likely non-osmotolerant, if you wanted to know the key term. Whereas for extreme halophiles, you'll have them grow up as concentrations of NaCl increase. This has relations to medical conditions as well, right? If you have, let's say, hypertension and a lot of NaCl, or sorry, you have like hypercalcemia or other like conditions where there's a high level of solutes in the bloodstream, some different bacteria or organisms are able to invade and make use of those environments readily. So for Helicobacterium salinarium or salinarum, rather, you can actually see the prefix salin or saline gives you an idea of like saline tubes, right? So it's able to grow at high saline conditions and therefore is more osmotolerant. So bear that in mind. Or like it has an affinity to grow at higher concentrations of solutes. That also depends on the solute itself. In the case of NaCl, you also have a halophile, right? So think about like, and that relates to chemistry, halogens, right? Because if you recall, NaCl, Cl, chlorine, iodine, those different types of halogens that connects to halophile, being able to essentially grow or be attracted to solute-rich or ion-rich environments. And then you can see here is a beautiful example with the halophile that it's able to go up, reach a peak at a specific concentration, and then go down thereafter. And so briefly, that's what you guys want to be able to understand, because these different terms, conditions, and these different classifications are noteworthy for your lab exam. So being able to know, or at least even outside of the lab exam, understand how these different concepts connect to many fields that you're probably interested in, be it medicine, laboratory pathobiology, research, academia, R&D, research and industry, or development, etc. And I believe I talked about the D value as well. One other point for the D value, I didn't mention this, but for your D value, it is the time that it takes to go from essentially one position in your colony or essentially in a population from one position to tenfold down. So it doesn't necessarily have to start from here. It could be somewhere in here, right? For example, and if it reaches tenfold under, let's say it's 6 times 10 to the power 7 here. If it goes down to 6 times 10 to the power 6, you want to be able to essentially take at the x-axis the difference between the two increments. So bear that in mind. I wanted to make it simpler for you to look at it from the very beginning and just compare when it goes 10 times down or tenfold down. But the idea is that if you were to get other values, and this is shown here, if you look from 10,000, right, shown on the y-axis of living bacteria, with a log at 4, and actually from 10,000 to 1,000, the two different increments are shown here. And you can see that at 10,000, it's the growth is at, or the position of the bacterial growth is at 27, let's say 27 minutes. And then at 1,000, it's actually at 40 minutes. So because it goes down 10,000 to 1,000, so 10 times lower, that difference in time is indicative of your D value. And so bear that in mind, folks, because that reflects how long it takes to go down tenfold in bacteria number or bacterial number, shown by CFUs, indirectly from PFUs. But the idea is that this difference was likely 13 minutes, would differ depending on the other condition, depending on like the temperature that you're dealing with or the other conditions that we've talked about thus far. Usually when we're referring to thermal death time or D reduction values, they refer to temperature. But in a nutshell, that's what you want to be able to derive from your graph and then provide, inshallah, all that data from what I've talked about for your post-lab. Any questions, folks? I'm going to go forward with this one more time. For your post-lab, actually, I'll provide it online. So that way you guys can hopefully get a 9-minute early start to leave. If you guys do want to stay, though, if you had questions, let me know. But once again, remember, you have a post-lab, right? Think about and describe everything that you've done thus far. Oh, and one other thing, record the values, the qualitative observations. We didn't do it today, but have a look at the other groups' data for the different conditions. You can look at theirs as well in the meantime before you leave. That'll be a simulation for you being able to observe the data accordingly. Well, so you only have sparse colonies here and then none for septomycin. Just to confirm.
Honestly, it was a little bit dense too, but it was like you could see the individual, whereas these ones are full.
Okay. Can you describe it? As, yeah.
Yeah, it says like for this one, all of them have that.
Thank you. No problem. Yes, and in the meantime, if you want to show me your figures from the post-lab of last week, you can. You can also check, just in case if you want me to look at it once again. I want to make sure you guys get it completed because technically this is your last week for a post-lab, right? Right? Because next week you won't have you will have a post-lab next week, but you won't have results to bring forward to like week 11. So bear that in mind. Bear in mind what I've talked about, folks. Just to briefly end off, post-lab includes everything that I've talked about, TDT values, D values across the temperatures. Make sure that if there's any outstanding figures, by the way, from the past few weeks, 10 weeks, if you haven't shown them to me, please do so. As well, to record the values that I've talked about for not just the viable counts, not just the absorbance readings, but the qualitative observations you've made across the different treatments that we've talked about. That includes temperature, pH, and salinity. Thank you so much, everyone. And I'll be happy to check anyone else's books in the meantime. Yes, I needed it.
Mohammed,
yes.
Sorry to interrupt. I just want to tell you the thing that.
Yes. Thank you. Folks, very important reminder before you leave, once again, make sure that you spray your benches with 70% ethanol. Make sure that you place back all your plates where they belong in the top left incubator. As well, please make sure, folks, that you've actually recorded the observations I made for across the other conditions as well.
I did want to save you guys the time to take photos, record those observations for yourselves, but you need to have them for the other conditions as well, for temperature, for salinity, and pH. So bear that in mind. You guys can stay in for a bit, but once your group is done, they need to bring them down to that end. So don't leave until your tubes are essentially placed back to where they belong. The cuvettes need to be rinsed. So once you're done, place them into the Westgadine. Folks, you need to all make sure that you do this before you leave. Place your cuvettes with the liquids or the cultures into the Westgadine. Rinse with 70% ethanol and invert into these white trays. Other trays should be brought back, all of them put together into that brownish tray for autoclaving. Please return back all of your trays. If you have questions or need to record the data, let me know. And please do this before you leave today. Any final questions?
Apparently at the draw. I don't even know what's going on. I think I'm gonna draw that. That's why I didn't draw it. It's describing words. Look at this. Let's see what it's like. Everything from that here. Yeah. And there was
So just as an update, there is an update to the viable counts. For those that want to record them for 50 degrees at 0 minutes, please do so. And that will help for your PEP for the plotting of your results from the microplate reader in your lab notebook as well.
No, it's just wrong. I'm yelling at her. Yeah. Ready? And not ready. Thank you. I'm also gonna ask you.
Could still have you in. Okay. I think I have you. Okay, no.
Yeah. The last weeks.
Okay. That's perfect. Folks, you can also send me on Quirkus your qualitative observations for the spec 20, your qualitative observations across the trays, so that way we could pull them all together as a lab for your post lab, and that's gonna be like your final post lab for the course. Yeah. Final questions. Yes. Give me one.
Sorry, this was just a result. Don't think
I
that.
understand.
Yeah, and then when there was also like this one, which obviously.
This is strong.
Yeah, I know that. But that's kind of what we were working with where I looked at the pictures.
That's okay. I think the one concern, the previous one, is that it could have been that it was.
Yeah, I can
mixed
see
up.
that.
Yeah.
Okay, I'll make sure to make a note on that in the previous. Okay, thank
That's
you.
perfect.
Yeah, I have a couple things. Oh, thank you.
I'll go. Is it okay if I
Yeah.
go? No. I didn't go through it. I wanna like
Go ahead, go back.
be fair. Okay. Oh, yes.
I didn't apply. I did think that I like even how you like today is like the last one is like how will you finish when you're how will you I've done everything I was going to do. I finished everything except for the past stay. That's the only thing I haven't been able to finish entire lab.
That's okay.
I just wanted to know like how it's so low if I've done all of that. I do my pre-lab, I do them questions, I do everything properly as well.
That was from, I believe, week 4 to 5.
Yeah.
'Cause